Welcome to Flagship Studio at JPM. This year's Nobel Prize in Physiology and Medicine was awarded to 3 scientists for their groundbreaking work on the human immune system. Discoveries that laid the foundation for what is now one of the hottest areas of drug development today, regulatory T-cell-centered therapeutics, in such areas as cancer, autoimmune disease, and transplantation. I'm Avak Kahvejian, General Partner at Flagship, and I have the honor of speaking with Nobel laureate Fred Ramsdell and one of the key scientists who was seminal in helping make some of these discoveries underpinning the prize, Roli Khattri. Thank you both for being with us. I wanted to start off by maybe giving people a sense for the immune system and how it recognizes self from not self. I think your discovery was obviously very important to that, but maybe give us a high-level overview of that and how things have really evolved.
You want to do it? I'll do it. So the immune system is obviously dedicated to protecting us from pathogens. That's its primary function, right? So we don't get viral infections, bacterial infections, fungus, et cetera. And it's very good at that. Most of us, go about our lives pretty well. But there has been recognized for over 100 years now the capacity of our immune system to recognize our own tissues and cause damage and cause problems. And so everybody probably has a relative or a friend who has Crohn's disease or psoriasis or some other autoimmune manifestation where the immune system recognizes our own tissues and attacks it and causes some form of pathology. Some are devastating, some are annoying, frankly, somewhere in between. And people recognized, again, 100 years ago that there is this balance, and somehow the immune system has to control itself. It has to be able to see virtually anything that's foreign to it. And so the immune system has receptors that it develops that can see thousands, millions of different things. And so there's the potential that some of those will look like our own tissues. And so we've known that that can be controlled for a long time. We didn't understand how. And so about 25 years ago, 30 years ago now, Shimon Sakaguchi, one of the co-laureates in Japan, identified a population of cells that seemed to be able to control the immune response and to dampen autoimmune responses. And what we did, so Mary Branco, the other co-laureate, and myself working at the same organization with Roli, identified the gene that controlled those cells. And so now we understand what the cells are and how the cells develop and how they control the immune response. And so we now have an ability to take what evolution has designed as the control mechanism for the immune system and figure out how we can manipulate that to our benefit.
Yeah. And the distinction between central tolerance and peripheral tolerance, can you touch on that?
Yeah, so all T cells develop in the thymus. I grew up in an environment where the thymus, well, T, which is for thymus, T stands for terrific and B stands for boring.
You're not biased.
Apologies to my antibody colleagues. But all T cells go through the thymus, or almost all T cells go through the thymus. And ones that recognize our tissues get deleted. They just die. And they're eliminated from the system. But it's an imperfect process. And so some cells that can see our own liver, skin, kidney, whatever, make it through. And so these other cells, these regulatory T cells that we've identified, are critical in controlling those. And in fact, there's genetic evidence that the cells that we've identified in this peripheral tolerance is at some level even more important to health and homeostasis than deletion in the thymus.
Interesting. So that's like the next frontier.
Yeah.
Yeah, and it was actually Shimon's work early on, before we found the gene, where he did some very elegant experiments taking the thymus out to really figure out when these Tregs are developing or enough of these have come out. And then once they are in the periphery, how they are going to be controlling the pathogenic T cells.
Yep.
And I think much after our discovery of the FOXP3 gene connecting it to Tregs, those were sort of developing in the thymus, but there is also Tregs that develop in the periphery much later. And that's kind of a field developed, I think, and FOXP3 gene discovery probably really helped being able to track those cells in the pathogenic situations, and playing a role in peripheral tolerance.
So let's rewind the time. We're back in 2001 timeframe, I think, where the FOXP3 mutation was noticed in
1998.
1998.
Yeah.
Okay.
Yeah.
And the mutation's noticed, and it causes mice to develop a certain type of phenotype. Why don't we talk a little bit about that, that links this mutation with autoimmune disease?
Well, so when I joined this company in '95, the company was founded by Bill Gates and Paul Allen, so two of the richest people in the world, right? And so they weren't there to make money; they were there to make a difference. And so they were willing to support things like genetic approaches to find nodes of biology that were important. And there's, aside from FOXP3, there's other examples of things that the company did at that point that have actually resulted in very valuable drugs and great insights into knowledge. And we had a collaboration with a group at Oak Ridge National Labs that had a mouse facility that was developed in the 1940s during the Manhattan Project. It was actually part of the Manhattan Project. And there was a mouse at this facility that had rampant autoimmune disease.
And they were irradiating mice, right?
They were, but oddly enough, this mouse was a control. It never was exposed to anything. It was a spontaneous mutation, so it's important to have luck on your side as well.
That's right.
And these people kept this strain alive for 40 years because they knew it was important, but they couldn't find the gene. And when we came along in the mid-90s, we now have the sequencing technology. We could find the gene. And so we spent 2.5 years including using a mouse room in a janitor's closet that we converted, I think illegally, and found the gene in 1998. But it was really the insight and the capability of this government lab to keep this thing alive for 40 years that allowed us to identify this gene. And it's really a testament to people's stubbornness to say, this is important. I don't have the tools to figure it out today, but we want to keep this going. And that was impressive.
Interesting. And Roli, you were working on this mouse and trying to identify the gene?
Yeah, well, the gene was identified with Mary Brankow's group, who was also at the company. I was trained as an immunologist, so I was really trying to connect it to the function because we knew Dr. Sakaguchi had identified these cells, suppressor T cells, and we had found this gene, and it's like, how do these two things connect?
And you weren't collaborating yet, or?
Not with Dr. Sakaguchi.
Nope.
And so a lot of the work of how do we connect this, and I think the group was also very smart that we made a lot of transgenic mice. And now that I think back about it, making those mice is both a commitment in terms of money and time. But having those tools really helped us make the connection and say, this is the gene that's controlling the function. And I kind of remember those times. It was exciting times.
Yeah. You mentioned money and time in the context of a biotechnology company.
That's right.
Fairly unique. So what was it that allowed that company to really do deep, thoughtful science like that and commit to something?
Well, some of it was Gates and Allen, right? And they really were committed, as I said, to making an impact, right? They wanted to change history, and they didn't want to make money. Now, I'm at JP Morgan, right? So that's not exactly the main focus of this meeting. But I think if you make a change and you change history, people will make money. I come up from the other direction. I think if you do something really good for people in biomedicine, people are going to make money on it. Yeah. And I think everyone recognizes that. Everyone's here to actually do something good for people. And they were willing to do that. And we had a different approach. And we said genetics was a powerful tool to teach us what was really important. And that was true at the time, and it is still true today. And I know so this is before we did have the human genome sequence. Right? We have that now. But that doesn't mean genetics is irrelevant. There is still a lot of genetics that will teach us and inform us what's important and how we can use that and transform that into therapies.
So that's a great segue. How did you make the leap from all the genetic discoveries of how the mouse is getting sick and make the leap to humans? So how did that leap happen?
So there are two components to that. One, as I'll say the first one, which is once we knew what the gene was, we looked for human populations that looked similar to the mouse. That is, young males who died early of rampant autoimmunity. And so we had some collaborations at the University of Washington and at Oregon Health Sciences, Hans Ochs and Bob Wilden, who had identified very rare families with exactly that phenotype. The males would die, the boys would die somewhere between a year or two of life and usually 20 years of life, unless they got massively immunosuppressed. And it was male inheritance, which was important for this gene.
Right.
And so we could easily say, we went in and asked, what's their FOXP3 gene look like? And it turns out it was mutated. But the other part of it was really understanding where the gene was expressed in either the mouse or the human, and it turns out to be the same types of cells and the same thing. And that was all the work that Roli did.
Yeah, and I think the fact that it was X-linked probably really helped us, right?
Absolutely.
Because there was another gene that everybody was thinking about in terms of this phenotype, CTLA4, and I came from Jeff Bluestone's lab, which worked extensively on CTLA4, and that's what we thought first. But knowing that it's X-linked kind of ruled it out to some extent.
Right.
So there is a luck component of it, but having the human phenotype and being able to find it in that population really helped us too.
Absolutely.
You know?
Yeah, so it sounds like FOXP3 mutation on the X chromosome points you to maybe male-oriented diseases, male genetics, and a clear phenotype to link that together.
Yep.
So maybe a unique situation, but not so unique, but allows you to make the translation more quickly.
Yeah.
For sure.
Right?
Yeah.
So how does that speak to the translatability of mouse observations, mouse studies, human studies, and other things like that?
So it was something you said during your Nobel lecture. It is, and I think all of us who are in this field completely agree, mouse is not human, but it is an extremely useful tool to point us in the direction we need to go into. The mouse disease is not going to look like the human disease. But the way you can really dissect out the details in a mouse and understand what your drug might be doing or what that pathway or what that gene might be doing, you can't really do it in a human population. Maybe a few decades down the road when machine learning has written all the algorithms connecting all the pathways to all the genes, maybe, but not today. I think mouse is still a very useful tool to kind of continuously come back and understand what's going on in the human biology or human disease pathway.
Yeah, the way I look at it is that a disease model in a mouse is almost always terrible. Because they're all acute. We give them disease, but in humans we've had disease for decades before we ever start thinking about therapeutics, for the most part. And even in tumors, tumors set up their own niches, et cetera. It's very different from a transplanted tumor in a mouse, very different. But the fundamental principles of how the immune system works is remarkably similar between humans and mice. And so if you're studying the fundamental principles of how the immune system functions, then I think the mouse is really good because you can poke it and prod it and you can do things to it and challenge it and ask what happens. Much harder to do in humans.
Yep.
And do you think you have to humanize the mouse? I know it's a technical question, but I know there's a lot of talk of mouse immune system versus human adopted immune system in the mouse.
I would say for most things, no.
For most things, no. Yeah. And the humanized system has its own problems and differences that even a humanized mouse is not anywhere close to human.
I mean, there's exceptions to this, but for studying principles of immunity, no. I don't think it's even useful. In fact, I think it could be misleading.
Interesting.
Yeah.
So you touched on already maybe 3 types of organizations that had to come together for this to happen, right? We had a public government lab, we have a private biotech company, we have universities, but we also now needed international collaborations. Tell us a little bit about how those international collaborations emerged. Was it at a conference, or how did the connection?
We actually never collaborated.
We have
Oh,
never, yeah.
interesting.
We've never collaborated. I mean, so you know, I've obviously spent a bunch of time with Shimon in the last 10 years, but we've never actually collaborated. And I think that's a great thing about the way the scientific ecosystem works, in that when he put out his information, we were able to use that. You know, Roli came into my office and said, "This is where this gene's expressed," and I was like, "Whoa, that's amazing. Okay, so now I have a much better understanding." But it was based on his information.
Right.
When our information got out, he was able to say, this gene is in my cells. But the rest of the scientific community also has participated in this. And so we have learned so much more about regulatory T cell biology and the way peripheral immune tolerance functions from the rest of the scientific community than we ever contributed to it. So we contributed the node that started it,
Yeah.
but the rest of the world has done the lion's share of the work.
Yeah.
And I'm going to sort of sidetrack a little bit and tell you why
Please.
we didn't continue doing it for two
sure.
different reasons.
Sure.
One was we were an antibody company, so looking for things that are on the cell surface. FOXP3, being a transcription factor, was inside the cell, so we obviously couldn't target it. We spent some time trying to do all kinds of things. Can we find the gene that FOXP3 controls so we could make an antibody to it? We spent, I think, maybe about a year or so. And at that point, we came to a place where, like, we can't really find just one single thing that we could make a drug against. And so as a company, as a small biotech, what we had to say was, we can't continue working on it. Doesn't justify. And we landed up then working with collaborators, sharing the findings, our tools and everything with Sasha Rudinsky, who's contributed an immense amount to the field, Jeff Bluestone, Steve Ziegler at University of Washington. So, you know, this back and forth between academic and industry sort of continued, where as a company, we couldn't take it to being a drug, and it went back into academics, and then now it has come back, and, you know, companies like Sonoma and other companies are taking it in the form of CAR Treg therapy. So I think there are discoveries that will go in one direction and come back to industry maybe sometimes later. Sometimes it's, you know, sometimes you're lucky in industry, you can continue working, but that's what happened to the FOXP3 and Treg project at that company.
Interesting. So maybe another sidetrack. Since it was done in the context of a biotech company, when was it published? When was it disseminated? I know there's a lot of secrecy and IP concerns.
Took a long time.
Took a long time.
Took a long time.
Yeah.
Longer than it should have, to be honest. But it was published in 2003. I think we knew the association in 1999.
Yeah.
And so that was, I would say, one of the great disappointments I've had, is it took so long. And it wasn't really because of the company. A little bit of it was, right? You're right. It took a while to file IP. But it also took a while for us to dot our I's and cross our T's and make sure we had the right biology. But it did take a while. And that's unfortunate, because it just slows everything down for reasons that it really didn't need to.
Yeah. So let's talk a little bit about the implications. You started to touch on that. Now that we have cell therapies, we have other alternative types of biologics, it seems like this concept now can reemerge. You don't need to target the transcription factors you had thought about.
Right.
You can now leverage this for other diseases and applications. So let's talk a little bit about that. So maybe you can start and give us a sense for where it's going.
Well, so currently where it's going is, I think the most promising is the CAR Tregs that, you know, companies like Sonoma are taking into the clinic. They are already in the clinic, have had some success initially. Actually, in terms of targeting FOXP3, the gene itself, I have tried it in my past lives. I'm sure you have too, and probably if the opportunity comes, maybe at Expedition Medicine too. Just again, a bit of a sidetrack, with covalent chemistry that allows you to target very specific proteins. we thought about targeting it, didn't have success in the past. We'll have to see what happens in the future. And there, the purpose would be to go into immuno-oncology, where you take away the Tregs from the tumor to make your immune system sort of destroy it. But that's a separate field. Right now, it's going into autoimmune indications. So the CAR-Treg from Sonoma is going into RA currently first, and then probably other indications. Will we have a way to amplify these Tregs, not through the Treg mechanism, by some other small molecule? I think that is a little ways away. Interestingly, in one of my past projects, not here, at Expedition, we did see, because Tregs are a balance of inflammation and kind of keeping the inflammation down. And what happens is when you keep down some of the inflammatory cytokines, we saw the Tregs kind of coming up naturally.
As a feedback.
As a part of the T cells doing what they do. When you damp them down in one side, they'll come out some other side. And in this case, it was coming out on the Treg side. So possibly it will come out, but direct targeting of FOXP3 is a little bit a ways.
Yeah.
Do you want to?
But you know, it was back in 2000. We said if you could put this gene into cells, or you could make a lot of these cells, we could treat autoimmunity. But no one was going to do that therapeutically. I mean, in 2000, CAR Ts in oncology weren't a thing in 2000. Yeah. That's right. So you know, you go 20 years later, technology changes, things advance, and now you have the opportunity to do this. And you know, will we find other ways to manipulate the system? I'm sure we will. Yeah. I have no doubt of that. How long and how efficient, I don't know. We'll find out. But at least we know what we're trying to do. And as the technologies come online, we can ask, is this the right technology to affect what we need to affect? And I'm hopeful and confident that that's going to continue to progress.
And so what are some of the modalities that are being brought to bear? You mentioned CAR-T. Is that ex vivo, in vivo, and then are there other ways? So
there's a number of companies. So I have one, obviously Sonoma Bio, but there's several other companies and some academic groups that are doing similar things, right? So they're adding specificity to the CAR, and they're doing it in the same way the oncology people do it. Take cells out, purify them, put whatever gene you want into them, grow them up, and put them back. And so it's a very similar, straightforward mechanism. And the question is, did you put the right genes in? Are you in the right patient population? Are you going in at the right stage of disease, et cetera? So there's still a lot of issues around this. This is version 1 of what we're trying to do. I think we're encouraged. I think a lot of groups are encouraged by this. But there are so many iterations of this and so many ways that I think will make this more efficient and effective. That I'm really hopeful. And I don't know, maybe we'll talk about this later, but the thing I really like about the cell therapy approach, which is really this kind of third pillar of medicine, Jeff Bluestone published this over a decade ago now, which is using cells to do something. And my version is, look, if we can use what evolution has designed or selected to maintain our health, I don't have to be smart. I don't even have to know how it does it. I just have to use the right cells. And so anytime I can make progress without being smart, I think that's good. So
says a Nobel laureate.
Right. Well, there's a lot of people, a lot of my friends who are much smarter than I am, and we still don't know how to make this work. So if we can use what evolution has selected to do what it's supposed to do, that's what I want to do. And again, we can talk about this. But the cells, yeah, they dampen inflammation. But they're in every tissue in our body, and they help maintain tissue homeostasis. They do tissue repair. They do a lot of other things that are less obvious, but potentially equally important. And so I think harnessing that is something that will happen over the next decade.
And you want to direct them to certain tissues, correct? So which or not?
I don't.
Okay.
You know, that's kind of this weird misconception. So regulatory T cells will infiltrate any inflamed tissue. Now, the ones that have specificity and see something in that tissue will stick around and do something. So what we do is we give them something to see that inflamed tissue. And everyone who's doing this, not just us, does the same basic thing. They give them the specificity for the transplanted liver or brain tissue or in our case, rheumatoid arthritis tissue, or whatever it is you're trying to diabetes, a beta cell antigen, whatever it is you're trying to do. You give them that specificity. Once they get there, they recognize it, they start doing what they're supposed to do.
I see. So they're perfusing everywhere.
They
and
perfuse
they're monitoring and looking for the antigen.
That's their job. That's what they do.
Yeah. And how about other approaches, using linkers or other types of things without the cell therapy component?
So the problem with this, as opposed to the bispecifics that people are trying to develop in oncology, there's nothing specific for a Treg. There is no marker on a regulatory T cell that you can target that will give you regulatory T cell specificity. So if you could find such a marker, that would open up a lot of things, and people have looked really hard, and there's just nothing out there to do that. There are other approaches. So people are giving low-dose interleukin-2. Antibodies to TNF receptor 2. There's other things that people are using to expand these cells in a sort of a non-specific manner. And there's some encouraging data in that. So, that may be another strategy to get at some of this. And I think, time will tell. The more people working on this, the better off we're going to be.
Yeah, and that's what ties into the indirect ways where you can enhance the Treg function.
Right.
But bringing Treg specificity is a little harder.
Yep.
So when are we going to see the first therapies, and in what areas? What do you think?
Approved?
Yeah.
I'm terrible at predicting the future.
I know.
I can tell you there's a number of companies and some academics that are still in clinic, in patients, seeing varying levels of encouraging data. Are there absolute cures today? Not that I'm aware of. Who knows? Maybe we'll hear about one later in this meeting, but not that I'm aware of. But we're going to get really good clinical data in the next 2 years, I would say. And that will tell us, are we really on the right track, and have the manipulations we've made been sufficient to give us a true, durable, effective therapy, or are we missing something? And if we're missing something, we in the community know many things we can do to augment these cells. We just don't know if we need to, and if we do, in what way. So I think in the next couple of years, we'll see an inflection point in how well we can manipulate the system and what we need to do to make it better. Now, for an approved drug, I will be stunned if it's in the next 24 months. But, drug development takes quite a while. We only started in the clinic a year ago or 18 months ago or something, right? So it's still pretty early days.
Yeah, but I think there will be a lot of learnings from these first set of clinical data. How long do you need to keep them? How much of it do you need,
Yeah.
So persistence, dose,
Persistence,
and I guess
dose,
efficacy.
and efficacy. Is there a certain disease better suited for this versus something, one of the others? It's
typical drug development, right? You start with the sickest people who've been on every drug possible and have not responded well to all the drugs, and now we're going to try another one. That's a high bar, right? So you sort of look for signals, and hopefully you can dissect who will benefit from this the most. There's very few panacea drugs. I don't know of any true magic bullet panacea drugs out there for everybody. So we need to figure out where this technology is going to contribute the best.
And in the beginning, going after places where there's a single antigen that you think is the most potent, or is it multiple antigens at the same time?
Right now, everyone is, I shouldn't say that. Most of the organizations that are manipulating the Tregs are going after a single antigen. But there are other groups that are trying to boost the entire system in a polyclonal, non-antigen specific manner. And again, they're seeing some intriguing data in that. And so, that may be, as I said earlier, I don't have to be smart. I'll let the cell do it. That may be simply more is what we need. Yeah. And in some cases, that might be sufficient, and this might be a way to get at it.
Great. Yeah. Maybe we can shift gears a little bit and talk a little bit about, Roli, how you're taking lessons from all of this and applying it in the context of your role at Expedition, right? In terms of leading a scientific team.
I think there are several. Some are more on the science side of things, and others were more personal learnings. You know, on the science side of things, kind of go in with curiosity and be willing to be wrong. That's, I think, one of Fred's favorite things. He said that's how he ended his Nobel lecture also, that I am wrong more often than not. And he had a story, very interesting one. But that's, you know, you have to be willing to be wrong and learn from it. I also have felt we did a good job of it at Darwin Molecular and subsequently having that industry academic collaboration to really leverage things that the academic labs can do and work with them closely to then enhance what you are doing for drug discovery. Another one that I learned, which I kind of already told you this story, is making a no-go decision. That was my first industry job. That was my first project leader role. The science was exciting. The field was like, had taken off, and we were having so much fun. It was a great team. And yet we had to bring it to a place where we had to say, we are not going to work on it anymore. That, as a young scientist, was a hard decision, but the right decision. So I think having that courage to make the decision of can't keep working on it as much fun as we are having. And then I think I would say on what I've learned from Fred in terms of mentorship, you saw an example of it. He's a Nobel laureate, but you saw his humility. And so that, I think, really helps bring a certain amount of trust to the team, where, like, if I go and tell my mentor, my boss, something that I disagree with, he's not going to be, I am right, you're wrong, that approach. He's never taken that. It really builds a trust in the team. And then I have a little bit of a personal story. Hopefully Fred doesn't mind going into it. It puts you in good light. So.
It doesn't matter. You're going to say it anyway, so it's fine.
So this is, when I worked with Fred is when I had my daughter, and I came back from my maternity leave, and he says to me, you're going to be the project leader for this FOXP3 project. And I looked at him, you know, anybody who's has an infant at home, comes back from maternity leave, knows you're in a brain fog. And I looked at him and said, Fred, I think these are the words I said, like, are you out of your mind? You're like setting me up for failure. But he's like, don't worry about it. I have your back. You'll do fine. So I think he was both, I would say, a cheerleader and a safety net. And that's like a balance that you have to play as a mentor. And it's an incident you can imagine. It has stayed in my mind and has had a huge impact on me. And I still tell this story whenever I'm lecturing students of building that sort of being a cheerleader and a safety net that your group can fall back on and knows that you have their back. I think that has been an incredible lesson that I've learned from you. So
that's great.
Yeah.
And Fred, why Roli? Why did you wait for her for such an important project?
Because she was really good at what she did. I mean, when we were building the team, we actually had a lot of choices because people were very excited to come work in an area where they had good support and were trying to do something pretty novel. And so we had a lot of options, but I know Jeff Bluestone, where Roli had worked, and I know Jeff's a terrific scientist, and I knew she would have been trained really well and knew immunology really well. So that was pretty easy. And then as a team player, when you're in I went into biotech because I liked the team environment of science. Again, it comes back to the fact that I could work with people who were going to be much better molecular biologists than I was ever going to be, or much better biochemists than I was ever going to be. And I could bring cell biology into the equation. So as a team, you can do a lot more than an individual in a lab, you know, doing collaborations, but really doing your own thing. There's trade-offs, obviously. But you had to have a team-like mentality, and Roli had that for sure. And you know, the people who worked with/for Roli would work with/for Roli again in a heartbeat. And you know, that's something, the ability to motivate people and bring people along and have them there on Sundays, and all of that is an incredibly valuable trait.
That's great.
Oh, thank you. I'm glad you asked him now, I know.
So just in closing, we all know of the late night fateful calls to Nobel laureates. So tell us your story.
Tell my story? Yeah, I've told this story so many times. So I go off grid all the time, and particularly in September. And so a little bit of background. I, with Sasha Rudinsky and Shimon Sakaguchi, won the Crayford Prize from the Karolinska Institute in Sweden in 2017 for this exact same thing. And that was an amazing prize, but it's given by the same institution. And I thought, well, okay, that was their way of saying, we like your stuff, but not quite enough. So but it was a great prize. We had such a good time, met the family. It was fantastic. And after that, never thought about the Nobel Prize. And so we were off in the mountains east of Yellowstone, well off grid, well off cell phone service, et cetera. It snowed. In fact, we almost didn't come out the next day. And we came out. We drove through Yellowstone. We saw elk and bison and everything you see in Yellowstone. It was fantastic. And eventually my wife's phone connected in some little town, and we stopped and pulled over, and I let the dogs out for a walk, and she screamed in the car. And I was like, what? And she comes out with this big smile and said, you just won the Nobel Prize. And I literally looked up and said, no, I didn't. And she's like, I have 200 text messages from our friends that said you won the Nobel Prize. I'm like, huh. I'm a scientist, that's data. Okay, I guess I won the Nobel Prize.
That's amazing.
And then we had another hour-long drive to get to a hotel where I could get service and get Wi-Fi and start calling people. And of course, I called the Nobel Committee back, and they'd gone to bed because it was 1 o'clock in the morning in Sweden. So I called and left a message and said, hi, this is Fred. I hear you're trying to contact me. Please call. And they did at 1:30 in the morning. And yeah, so I was mentioning earlier, I think there's literally one phone call that anyone gets between midnight and 6 a.m. that's good. Right? Normally it's your kid's in trouble, or somebody died, or something bad has happened. And this is like the one call that literally 1,000 people in the history of the world have gotten between those 2 hours, and I missed it.
Yeah, yeah.
It's very disappointing. But my donation to the Nobel Museum was my hiking boots.
That's funny. All right, that's great. Well, look, thank you both for sharing such a candid version of the science and the history and your relationship. That was really, really, really informative. Thank you very much.
My pleasure.
Yeah.
And thank you all for joining us at Flagship Studio at JPM.